Good afternoon, Tridev. Okay, we will just wait for a couple of more minutes before we start. I'm not sure or not because it says that um, the network is kind of unstable. Okay, so all right, I think we are back online. In today's class, we will look at an important concept of how to introduce foreign genes, right? We have talked about cloning, we have talked about using plasmids, and, we talked about cloning vectors, expression vectors, and all that, right? After having all that, we need to introduce these transgenes into appropriate system. Could be bacterial cells, could be plant cells, could be animal cells, could be yeast cells, Somehow we need to introduce these cloned DNA into those organisms. Okay, so in today's class, we'll spend some time looking at, the different protocols or different ways by which we can introduce these foreign genes into the organisms. Let me share my slide. Um, no, this thing videos today. All right, I'm sharing my screen. I guess my screen should be visible now. All right, there we go. Okay. Okay, so the first thing that we are going to look at is bacterial cells, right? How do we make sure that the bacterial cells are taking up your DNA? Okay, typically, if you remember your early school, we all knew how this, S phenotype of pneumococcus became R phenotype. Simply, the bacteria took up the DNA and R became S or S became R. It can happen either way, right? So we all know that the bacteria have the ability to take up foreign DNA, and that process is called as transformation. Okay, so different bacteria have different ability to take up DNA. Some bacteria naturally take up, DNA, and this process is a big nuisance because it results in what is called as horizontal gene transfer, and that is actually responsible for the antimicrobial resistance. For example, when you take any antibiotic, when you take those antibiotics, the cells will lyse, they will, the DNA gets digested, and at some point some of these fragments are picked up by different DNA by different bacterial species, and sometimes it could be coding for antibiotic resistance. So antibiotic resistance is now one major challenge that clinicians are facing because no new antibiotics are being discovered, and antibiotics that we are already using, many bacterial species have developed resistance to it. So it does look like a catastrophe when we look at the antibacterial development, because we barely have anything in the antibiotic pipeline. Okay. So this process of bacteria taking up exogenous DNA, right, not endogenous DNA, exogenous DNA is has a problem. Okay, and bacteria like Bacillus subtilis can take up DNA naturally, but there are other bacteria which do not take up DNA fragments naturally, but you can do certain processes and make sure that these bacteria which do not take up naturally, we can modify them so that they will take up DNA. Okay, and that process is called as competence. So we make cells competent so that they can take up the DNA. Okay, and competent cells can be either treated with electric shock so that they will take up the exogenous DNA, or they can be treated in certain ways so that they will take up the exogenous DNA. Okay, so we'll start with bacterial transformation. Okay. Um, when we look at bacterial transformation, both gram positive and gram negative bacteria have this ability to take up exogenous DNA. Okay, and there is definitely, each bacterial species has its own capacity, right? Whatever fragment exogenous DNA it takes, it recombines with the host gene in regions where there is homology. It doesn't go and integrate randomly. Okay, it basically looks for homology, and it goes and recombines very close to that site where there is a lot of homology, and this process is mediated by a series of proteins called RET-A, RET-B, and RET-C. Okay, these are three genes that are responsible for making sure that the foreign gene, this one depicted here in orange, is picked up. Is You will see a base pairing along those complementary regions, and then eventually, this replaces the original strand. This gets degraded. Okay, and you will have one in which this. So this is just showing you a single strand. Imagine it's double stranded. Only one strand is going to get changed. Okay, one of the other strand will not be. Okay, and so you will actually have a partial diploid. Okay, so you will have original and the recombined this thing. Okay, so 50% of the progeny will have the recombined one, and 50% will have the original one. Okay, and this is one important process in the entire recombinant DNA technology, where we have this ability to introduce foreign genes and express these foreign genes at any point, right? So E. coli has turned out to be one major workhorse or one major model organism in the expression of foreign genes. Okay. So bacteria like E. coli, they don't take up DNA naturally, but they can be, treated with certain chemicals like calcium chloride, right? So when you treat E. coli cells with calcium chloride, keep them at low temperature, they have this ability to take up plasmid DNA into the cell. Okay, and the eventually the plasmid DNA is stably propagated in E. coli, and we can make sure that our recombinant protein or whatever we have cloned into that plasmid will get expressed. Okay, as we saw in our lectures in cloning and expression vectors, depending on whether the gene is being put into cloning vector or into an expression vector, the host will be different. Okay, the host for cloning and expressions are different, but nevertheless, the other processes remain same. For example, in this picture, what it shows is that the surface of E. coli is, full of what is called as LPS or lipopolysaccharides, right? So when we treat these bacterial cells that are growing logarithmically with calcium chloride and leave them at as at a very low temperature, typically up to 4 degrees centigrade. When you keep them on ice, within the membrane, you will have regions, okay, in which the plasmid can be taken up, okay, and this itself is mediated by a series of proteins, okay. These are called as competence factors. Okay. They bind to this DNA, and then once they have bound, the moment you give them a heat shock, right? So until all these things are at 0 degrees centigrade, you suddenly shock them at 42 degrees centigrade. So going from 0 to 42 is a heat shock. This entire process gets completed. That means the DNA gets internalized. Here, as you can see, as part of it is internalized and part of it is externalized. A lot of it is still in the external surface, okay. The moment you give heat shock, the entire DNA gets internalized, and then after, you don't give heat shock for too long. You basically give only a pulse. So, you know, you give heat shock for anywhere between 30 seconds to 90 seconds. Okay. Some people give it for 30 seconds, some people give it for a minute, some people up to 90 seconds. Nothing beyond that. So in about anywhere between 30 to 90 seconds, this DNA gets internalized, and then at the end of heat shock, you will be growing them at 37 degrees centigrade, and this plasmid now, establishes itself within the bacteria. As we know, some plasmids are called as high copy number plasmids. There can be as many as 500 copies per cell, and there are some which are called as low copy number. That means anywhere between 5 to 10. Okay, only that many plasmids will be present. So PUC, for example, PUC 18 and 19. Sir? Okay, sir. Okay, sir. Okay. Thank you so much. Thank you. So PUC 18 and 19, for example, are examples of high copy plasmids. Okay, and PBR 322, for example, is a low copy plasmid. Okay, so within a cell, when we try to transform with PBR 322, about 50 copies at the maximum, will be there. On the other hand, things like PUC 18 and 19, you can have as many as 200 copies of the plasmid can be there. Okay, 200 numbers. So when we try to make a plasmid prep, you will get lots of DNA when it is on a PUC based vector rather than on a PBR 322 based vector. Okay. So this transformation is something that is routinely used, and it's a good way. It's a good way of introducing foreign genes into bacteria. So how do you make things like E. coli take up the foreign DNA? Simply by treating them with calcium chloride. Leaving everything on ice, and then do a heat shock. This is literally a I won't say fail proof, but by and large, it's a very robust process. And if I were to tell the you students, if I give these you students how to do this, you will be able to do it pretty well. Okay. So that means the process is robust. Okay, in my hands, I may get maybe 100 transformants. When you people do, you may get about 50, or maybe less than 50 transformants. But nevertheless, the process works really well. Okay. There are other ways other than there are certain bacteria which do not take up DNA naturally and which cannot be made competent by calcium chloride. Okay, in these cases, we can use what is called as electroporation. Okay, so it is an instrument-based system which pulses, which gives very short, very brief, microseconds, gives a high electric shock, and because of that, your double membrane, your membrane bilayer gets ruptured randomly. Okay, and because these cells are floating in a solution that has lot of DNA, the DNA gets inside the cell. Okay, so you give a brief pulse and then switch it off. So as I said, it's typically lasts for about a microsecond, and once the membrane layer tries to reseal, invariably takes in the contents of the outside medium also. So the DNA gets in, and once the DNA gets in, it starts expressing, and you can have a electroporation is another means by which you can use foreign DNA. Okay. Now, when it comes to yeast, for example, instead of calcium chloride, people use lithium chloride. Okay, it does look like lithium chloride is much better than calcium chloride for yeast. So people have stuck to lithium chloride, and it works fairly well for yeast cells. Okay. When it comes to mammalian cells, people realize that there are at least this process of introducing a foreign gene can be classified into two. One is instrument based, okay, and the other is reagent based. So, in instrument based, you're going to use some instrument to introduce the foreign gene into the organism. Could be E. coli, could be yeast, could be mammalian cells, okay. And when it is reagent based, uh, by and large, you don't need to use instrument. Instead, the reagent has this ability to take the DNA into the cell. And then now you have a combination where you use a combination of reagent and instrument. I will show you an example towards the end. And you can use a combination of reagent and instrument, and you can improve upon the delivery of this foreign DNA into cells. But why do we even need to deliver foreign DNA? Simply because we want some of these proteins to be expressed. Some of these proteins could have a therapeutic value. Some of these proteins could have, could be acting as an antigen. So, you need to purify it, express it, and purify it, right? So there are different methods. Okay. So typically, the other way we can classify the route of foreign gene introduction is by naming the methods. Is it a physical method? That means where you have electroporation, gene guns, you have micro injection, you have micro projectiles. It's these are tiny, missile-like things, are tiny particles that can be, you can bombard these cells. It could be a plant cell, could be an animal cell. When you bombard, you're basically introducing a small hole at the site of impact, and the foreign DNA, which is along with those particles, will get into the cell, and you can express the foreign gene. Okay, you have chemical method. For example, there are chemical methods where we can use certain chemicals and mix them, and then use these chemicals plus DNA combination. When you incubate with cells, the cells take it up. For example, there's a thing called DEAE dextran, which has this ability to bind to DNA. Okay, and once you transfer, it's easy you make a complex of DA dextran and your DNA, and when you incubate with mammalian cells will take it up inside the cell. Inside the cell, DA dextran and DNA come out, and the DNA can be propagated naturally. Okay. Instead of DA dextran, you can use calcium phosphate. Calcium phosphate has, this phosphonate ability to bind to DNA, okay, and all cells need calcium, right? So calcium phosphate also has this ability to get inside the DNA. Okay, so you can into the cell, into the nucleus, and so you can actually have, you can complex your DNA along with calcium phosphate, and it gets delivered into the cell. You also have lipids. There are certain liposomal preparations. Okay, liposomes are nothing but chemically modified lipids. Okay, chemically modified lipids. They have this ability to bind to DNA. Okay, they have this ability to bind to DNA, and these can help the cells to fuse, and the lipids will be internalized, and the transgene can be expressed. Okay. The third category is biological, which means you can use viruses. You can use viruses as vehicles for expressing a foreign gene. For example, in the current pandemic, one of the COVID vaccine, right? One of the COVID vaccine is nothing but it's a recombinant adeno-associated virus that displays the particles on the surface. So you can have, virus-based gene transfer methods. People knew, for example, things like adenovirus, adeno-associated virus, retroviruses. They all have cell preferences. That means, they will infect only certain cell types, and you can use these to introduce foreign genes into any cell type, based on the kind of virus that we're using. For example, HIV, we all know that it infects only CD4 positive cell, nothing else. HIV will not infect any other cell. Generally, it infects only CD4 positive cells. So, you can have a very, very selective payload delivery. That means, your DNA can be delivered very selectively by the use of certain reagents or viruses, or by use of instruments. Okay. What we call as transformation, the introduction of foreign DNA into bacteria, is called as transfection in mammalian cells. So in reality, transformation and transfection are nothing but a process by which we are introducing foreign genes into either into a bacterial cell or into a mammalian cell. But for whatever reason, for historical reason, this is called as transfection. When it comes to mammalian cells, it is called as transfection. When it comes to bacterial cells, people call it as transformation. Okay. Now, transfection itself can be classified into two major categories. One is called as a transient transfection. Okay. The other is called as stable. Okay. So, transient is when genes get expressed for a very limited time. Okay, so this is again, depends on the vector that we are using for expressing the gene of interest. Okay, the transgene expression is very high. Okay, and there is no need for selection. Remember the cloning vectors, expression vectors, we said we need to select for ampicillin, we need to select for certain antibiotics. There is no need for selection when it comes to transient transfection in mammalian cells. Okay, and invariably, these plasmids are propagated as episomes, and over a period of certain this thing, the episomes can be lost. Okay, can be lost. Okay, need not be, but can be lost. Okay, in stable transfection, genes continue to express beyond 72 hours, and the expression is very high to begin with, and then it starts to become lower and lower and lower. Okay, and in stable expression, selection markers are used, And in general, the reason why we call it a stable expression is because it is integrated into the genome. Okay, so you have episomal expression in the transient, but when it comes to stable, it is because of integration into the genome. Okay, any questions? Okay. That's how the picture looks like. That's how the picture looks like. Stable transient transfection. This is a mammalian cell line that has been transfected with green fluorescent protein. So you can actually see in transient transfection, literally, more than 90% of the cells have taken up the green fluorescent protein, and it fluoresces very brightly. Okay. On the other hand, when it comes to stable expression, there are very few cells that are expressing at that high level, but then they will continue to express. They will continue to express at a very high level. Okay, but the fraction of cells that are going to be stably expressing is kind of low. Okay, and you need to select for them. You don't know which cells are going to be stably expressing, so you may need to do a selection process. Okay. Okay. So electroporation in mammalian cells. Okay. So exposure to high intensity electric field during the electroporation destabilizes the plasma membrane. At this point, the destabilized plasma membrane is more permeable to molecules that are present in the external media, and DNA is taken up along with other molecules in the media. And after the field is turned off, the membrane returns to its original state. That means these pulses are going to be extremely brief pulses. Okay, for example, I said that we give heat shock to E. coli for about anywhere between 30 seconds to 90 seconds, right? On the other hand, when it comes to mammalian cells, when we are doing electroporation, these are of the order of, few nanoseconds. Okay, few nanosecond, and the moment field is turned off, all this DNA, everything wants to rush into the cell. Okay. Now, two types of electric fields are used. One is called as exponential, other is called square waveform. Okay. So both exponential decay and square waveform have been used for transfection. It is simply because some cells seem to respond very well when square waveform is used. Okay. On the other hand, in certain cell types, only exponential thing works really well. Okay. So it all depends on the cell type. So this is just shows. So these are the cuvettes that are used for electroporation. Okay, so this is how the cell looks like before it was pulsed. Right. So you have the outer milieu, the outer environment of the cell where you have these different DNAs. Okay, and there's the binding, and these DNA, when we give a short pulse, what you can see is create small pores, and the DNA gets in. Right? As long as your membrane integrity is not lost, everything remains on the outside. The moment it is given a heat shock, then these DNA will get into the cell, and then it will start expressing, and then you can select for those expressions. So essentially, electric field induces, a short pole, and the DNA can enter it. You have two other methods. One is called as Helios gene gun, okay, and the other is called as biolistic particle delivery. Okay, both of these are undergoing trials. Okay, Helios gene gun has been used successfully for gene delivery to animal, plant, cell culture, nematodes, yeast, bacteria, all these different things. Okay, it is used based on the pressure sensing capability. Okay, so it has it is able to generate huge pressure, and that is what is used for introducing the foreign gene. Okay, this can be this instrument Helios gene gun is highly portable, so you can take it to any field. Only thing is it needs a smart charge. Okay, So people are developing advanced version of the stereoscope. Then you have bioelectric particle delivery system, which is pretty much the same. Only thing is the pressure range is different. The pressure range is much higher here. Okay, and this generally doesn't work very well for certain big organisms, and certain other this thing. But for mammalian cells, it's okay. For expression, it is still okay. Okay. There was this another technology which was called as nucleofection. Okay, this technology was developed in the late 90s and came to market in the early 2000s as an efficient non-viral method for transfecting both primary and hard to transfect mammalian cells. Some mammalian cells are really difficult to do this. Okay. Hello. Anuj, I am in the meeting. Can I call you in about half an hour? Oh, okay. So this okay. So this nucleofection technology is based on the momentary creation of a small pore in the cell membrane by an applied electric field, and this is a highly customized setup. Okay, that means suppose you are using some cell line that is popular among scientists, then you may not have much of a problem. But suppose you're using a cell line that is rarely used by the scientists, but because of your work, because of the nature of your work, you have to work with that cell line. You may have to do certain customizations. Okay, and typically when you look at transformation, around anywhere between 70 to 90%. Here, with this, you can grade efficiency is greater than 95%, and it basically remains constant. Okay. Unless the solution has gone bad, then or the cells have gone bad, then it becomes difficult. These are the different modules that are available for nucleofection. So this is a commercially available device. Okay, when we talk about DE, Dextran, or any of those technologies, they may not be such hi-fi, but they work very well. They work very well. Okay, so it depends on what your purpose is and all that. So in this case, it shows what all how many cells can be transfected. The cuvettes for nucleofection also kind of special. Okay, cuvettes have this ability to conduct very high voltages. Very high voltages. Typically, the voltages supplied can range anywhere between 1200 to 1500 volts. The household electricity is around 220 volts. Okay, so when we expose these cells to an intense field, which is like almost 10 times higher, then typically the it results in the creation of pores, and some cells will take up. So many a times when you use these cells, depending on how well you have grown the cell, how happy the cells are, sometimes the entire cell may be destroyed during the process of either heat shock or when you give electric shock. So we may have to start all over again. So we have to be extremely careful when we plan for our experiments or whatever with these organisms. Okay, so nucleofection essentially gives high transfection efficiencies across many different cell lines. Okay, some of these cell lines are really hard to transfect using conventional technologies, but with nucleofection, you can have very high, transformation or transfection efficiencies. For example, you can see it's all in the high 90s. Okay, human T cells can be transfected at very high efficiency. Similarly, rat hippocampal neurons can be transfected at very high efficiency. Okay. So this just shows, many different cell types or cell lines were used, and they had this maximal response, right? Most of the cells, more than 90% of the cells took up the foreign DNA. So nuclear is something, it's kind of costly because you're going to customize it. Even if you're using a cell line that is commonly used by other researchers, invariably, typically what happens is, uh, sometimes these processes really work very well, and because it's a commercial company, nuclear fiction, you will have to buy all the reagents from them. Okay, so in that sense, it's kind of costly, and but you have to understand that this is highly efficient. If you're working with a cell that can be transfected very easily, you don't have an issue. But if you're working with a cell line which is very, very resistant to transfection for whatever reason, transfection doesn't happen really well, then nucleofiction is the way to go. Okay. The other processes by which foreign genes can be introduced are things like microinjection, laser fection, etc. Okay, so what is the microinjection? You basically have two things in microinjection. One is a vacuum line, okay, vacuum line, and the other is a extremely fine glass needle. Okay, so you basically hold the tip, this tip which gives you a small subtle vacuum. You will be using either an egg or any other cell type depending on the cell type. Some cell types have this ability to adhere. They'll go and adhere. Some cells have the ability to stay in suspension. So depending on which one you're using, you can resuspend it and you can do the micro injection. Okay. In the other one, one is this vacuum to make sure the cells are held firmly. The other is a small needle, extremely thin needle. Okay. Mostly people who do micro injection have a machine or a machinery that can make fine needles, extremely fine needles. Okay, your DNA mixture, everything is buffer, everything will be in this micro needle, and then this micro needle, then once it is injected into the cell, okay, with a small push, force, you can empty contents of this micro needle into the cell, and the cell will become transgenic. Okay. Similarly, you can coat your whatever you want to deliver. It could be DNA, it could be RNA, it could be protein. You can coat it to gold nanoparticles, right? Gold nanoparticles, and then when you shine, a laser at 532 nanometer, just as what happens with electroporation, you have tiny holes being generated because of laser. Okay, and these cells, this mixture of gold nanoparticles and your recombinant DNA can easily enter into a cell. All these things, anything that happens to the membrane is extremely transient, doesn't stay like that for too long. Otherwise, cells will die, right? So typically, people choose, scientists choose what strength the electric field should be, how long should people give electric shock. All these things are chosen. Okay, most of them are available in the literature. Even if it is not available in the literature, you can try various combinations and then see which one works really well. Okay, so depends on the cell type. So this is something that we had seen during our cloning vector lecture. This is a cloning vector. It's called PCMB6. This is typically used for mammalian transfections. Okay, it has a CMB promoter. It has a multiple cloning site with few restriction enzymes. It has a poly A signal, and it has a this entire gene is promoted by what is called a CMB promoter, cytomegalovirus promoter, which is one of the strongest promoters. Okay, So this is one vector. You have another vector. This again we saw in our cloning vector lecture. This is another vector. This is called as a retroviral vector. So this has an element of retrovirus in it. Typically, if you use these retroviral vectors, the idea is for it to integrate into the genome so that you can have a stable expression. For example, all of us know that HIV integrates into the host genome, and that is why people relapse, after several years, right? So it is because it is sitting there inside the genome. Okay? So this just shows one example of a retroviral vector. The other methods are, you can use biological methods or retroviruses or DNA viruses can be used for introducing foreign genes. Okay. For example, retroviruses, you have urine leukemia virus, MUMLV, okay, and you have HIV, you have HTLV. In DNA viruses, you have adenovirus, you have adeno-associated virus, you have HSV or herpes simplex virus. Okay, all these different viruses have certain non-essential genes in the genome. The non-essential gene can be removed and you can put your foreign gene of interest. Okay. Okay, adeno-associated virus, each of these have specific property. For example, adeno-associated virus are single-stranded DNA virus that specifically insert their DNA chromosome 19. Okay, so it seems to have certain, special affinity for chromosome 19 DNA. Okay, and herpes simplex virus, it's a double-strand virus with affinity for neurons, not for any other cell type, right? So it seems to do, transfer this into neurons. So preferentially infects neurons. Okay, so these are properties of various viral vectors. As you can see, the size, of the plasmid ranges anywhere between 5 kilobase pairs to almost as high as 40 kilobase pairs. Okay, herpes simplex virus is about 40. It can take up to 40 kilobase pairs. Okay, and as we saw, herpes simplex virus has this ability to infect neuronal cells, which are typically non-dividing cells. Dividing cells, it's easy. Non-dividing cells, as a general rule, it's a tough one to transfer. Okay, and as you can see, some of them are stable, some of them are transient. Okay, and but each one has a some kind of a disadvantage also. So there are advantages, there are also disadvantages. Okay, so when it comes to introducing a foreign gene, typically, I'm just showing you what happens with mammalian cells. Okay, you can imagine what would happen with bacterial cell. Bacterial cell, as I said, is treated with calcium chloride as it is growing logarithmically, and then you can make it to you can induce it to take up the foreign DNA. On the other hand, you have things like agrobacterium, and agrobacterium has certain plasmids called T plasmids, in which you can clone your gene of interest, and then agrobacterium can be used to transfer this gene of interest along with its DNA into the roots, okay, or shoots, whatever, depending on the callus, whatever you have made, agrobacterium has this ability to infect those things and it can transfer the DNA. I'm not, adept in plant cell culture, so hopefully you will get to hear more on that from Dr. Sunny, I guess. Okay. So in the case of mammalian cells, for example, if this is the expression vector and if these are the cells that are going to be transfected, in this case a cell line called 293, HEK293, human embryonic kidney cells, HEK293 cells, okay, with the gene of, whatever you want to express it, in this vector, can be introduced into this HEK293 cell lines, okay. After you plate them, when you plate them, about 5 to 10% of the cells will get infected and they will produce more progeny, right? So you can either go for continued expression or you can collect the virus and then go for another culture. So now it, mix this reagent. It comes with a specific reagent. Okay. Mix this reagent and the cells. Okay, so cells and this expression vector will be coupled or will be complex to a reagent. These two are mixed together. Okay, and you can infect, and the cells will start. Maybe in about 2 to 3 days, depending on the cell type, depending on the virus. In about 2 to 3 days, the transgene starts getting expressed, and you will see the cell line expressing the protein of interest. The ultimate aim in all these things is for the expression of foreign gene. Okay. Suppose you want to, say express an insulin gene, or suppose you want to express the spike protein of coronavirus. All you need to do is put this in expression vector. Make sure that you have this itself, is accompanied by a buffer and few other things. Along with the cell, you can start making, the spike protein or whatever protein that you want in the mammalian cells. Okay, so this is something that I just thought I should share with you, and I will mail you the slides. Okay, and so we are now pretty much close to finishing 2 modules. Okay. Um, I remember I told you that we need to make assignment. There is a home assignment and there is a group presentation. I would rather think that let's concentrate on home assignment. Just remind me, Devanshi, I would like you to remind me in the evening. I will mail out the assignment details by today evening. Okay, and let's turn in the assignments positively in 2 weeks time. Okay, which means by the week of October 20, 21, 22, one of those days, let's turn the assignments in. We need to plan for our midterm exams. Devanshi, again, check with the entire class and then let us see if we can do this test over a weekend. I don't want to waste my class. We can do this in a weekend. Okay?
Okay, sir. Okay, sir.
All right. Any questions on today's this thing? I will share this. Any questions? Okay.
No, sir.
Right. If there are no questions, then I will stop my presentation and then we will meet on Friday then. All right?
Okay, sir. Thank you, sir.
Okay.